Hello, thank you for joining us today in GHGA Webinar. Topic today is benchmarking and quality control for genomic variant calling with Johannes Köster. For those of you that they are new in GHGA, I will to say we have also other events. We have also a lecture series. You can join, for example, next week. We have a talk with Antje Kepler, the Euro Bioimaging Biohub and its imaging services. You can sign for that in our webpage, ghga.de, under events, or just scan the QR code. We also are organizing a summer school. The topic is gen set enrichment analysis with R. And you can also join us. You find future information in our website. We are GHGA. GHGA means the German Human Genome-Phenome Archive, and we are a data portal. We are the safe house for genomics, and we enable a safety way to share genomics for research use, and in doing so, we support we support research, and we support fundings and work like Johannes is doing. And that's the key word for today. Johannes is our speaker. I will briefly introduce Johannes. He made his PhD in genome informatics in the University of Dortmund, and then he was postdoctoral researcher for life science at Dana-Farber Cancer Institute and Harvard University and CVI in Amsterdam. And he is a group leader for he was a group leader for algorithms for reproductive bioinformatics at the University Duisburg-Essen, and now he's full professor of bioinformatics and computational oncology at the University Duisburg-Essen. I'm not extending this anymore. Johannes, the floor is yours. I give over to you. Thank you for being here. It's a pleasure.
Yeah, thanks a lot for inviting me and for the organization. Okay, so this talk will be about benchmarking and quality control for genomic variant calling. And I thought I might start with some basics about genomic variant calling at all and why there is actually a need for benchmarking in this field. So as I guess most of you will anyway know, but still let me briefly recapitulate what this is about. So genomic sequencing usually takes the DNA and then it generates small or not so small anymore nowadays fragments, depending on the technology of DNA, and each of these fragments is sequenced. Sometimes they are paired, sometimes they are single-ended sequenced. And what you get then as a computer scientist, so as a bioinformatician, is basically lots of small texts like these here over the alphabet A, C, G, T for the 4 nucleotides. And this basically contains the information about so-called genomic variants, and thereby genomic variants are differences in the individual's genome compared to some reference genome. And there's the reference genome for humans is like known, I don't know, since like 1990 or so, in part at least, and nowadays, we are pretty sure about almost every single position in this reference genome, but this does not mean that you can be pretty sure about every single genomic position in the reference genome, in the genome of a patient. And for this, you need to basically decipher these individual so-called sequencing read information into what is actually a variant or not. And the first step in that process is to align those sequencing reads against the given reference genome. So imagine that this black text here at the bottom is the reference genome, and the individual shorter texts here are these individual sequencing read. And then this alignment step is basically trying to find the position of origin for each of these reads in this long reference genome. Okay. And then the challenge is basically to understand whenever there is a difference in the individual reads against the reference genome, whether that is actually a genomic variant, so really something where the patient or the individual's genome differs from the reference, where there is something in the individual that might differ from many others in the population, for example, or whether such a difference is actually a sequencing artifact, so something that occurred while determining these short texts here. And this distinguishing between these two kinds of signals, so artifacts and true variants, is called variant calling. And the challenge here, as mentioned, is basically to understand what is a variant and what is actually an artifact. And this usually boils down to making certain assumptions about the individual's genome, like, for example, in case of the human species, that the genome is usually diploid, as you can see here at the top, in the sense of that every chromosome occurs in a pair, or at least most of the chromosomes, except the sex chromosomes, occur in pairs, and therefore, any kind of mutation or specific variant has to be either in one of the genome copies or in both of them, meaning that if you see something like this here and it's present in roughly half of the reads, this can be an indication of a variant that is only present in one chromosome copy, whereas if all of the reads would contain this alteration here, it would be like a so-called homozygous variant where basically both chromosome copies have the variant. And in contrast to that, sequencing errors or artifacts usually occur at other frequencies, like only in single reads. That's pretty simple, or only in a subset of the reads that is far below this, being 50% or 100%. That's the simple case, or the comparably simple case. I will come to problems with even that later on. But there is also more complex cases where you're not interested in so-called germline variants that are present in all the cells of an individual. But instead, you want to see somatic mutations, so stuff that occurs like during the lifetime of an individual, like, for example, which is very important and interesting for samples from cancer tissues, for example. And there, you cannot expect everything to be either 50% or 100% of the reads that you obtain, but there can be lower fractions as well. And then it's even harder to distinguish them from sequencing artifacts. Okay. So now the sequencing process and also the analysis of the sequencing data, so basically everything that goes from here to here at the bottom, is containing lots of uncertainties or sources of uncertainties. And I would like to start with the actual sequencing. So nowadays we have two dominating technologies. One is short read sequencing, like you get with Illumina sequencing, for example. This is basically fluorescence label based. So basically every nucleotide in the sequenced DNA gets a colored label, and these labels are basically attached via so-called sequencing biosynthesis to the investigated DNA fragments, and then basically the device takes an image of that and basically tries to distinguish the different colors of the different labels, and by that determine what nucleotide is observed at each position of each DNA fragment that is investigated. And in contrast, long read technologies exist. One of them is the nanopore technology, and there the DNA as a molecule a typically unfragmented molecule is pulled through a tiny little pore in the nanoscale. And while traveling through that pore, the current is measured. And by the differences in the current, you can determine which nucleotide has traveled through the pore at that point in time. So you see that like that, you get signals like this here. And both technologies have issues with uncertainty or certainty of the measurements. So here you can have, let's say, somehow weak or distorted signals of the fluorescency, and thereby sometimes you're not entirely sure about the individual nucleotide that is called at a particular position of the read. And here it can be that the current measurement is not as exact, and sometimes the solution is not as unambiguous as it looks here, and sometimes there are therefore differences. And in particular with this nanopore stuff, an additional source of ambiguity is that the travel speed through the pore actually can have an influence on the signal, and that is most important in case of seeing so-called homopolymers, so where you have multiple nucleotides of the same kind just next to each other. Then it's sometimes unclear whether you see like 3 or 4 or 5 of the same nucleotide or less. The same kind of errors also can occur with Illumina sequencing, but they are not introduced by the measurement process, but rather by PCR amplification, because that can make errors in these kind of regions as well. Okay, so the next step that can cause uncertainty is the alignment of the reads. So as mentioned, for every of these short texts, we want to find the position in the reference genome. And there, it depends a lot on the reference genome structure, whether it's always clear where a certain text or read is belonging to. And that can be hampered by, for example, repetitive regions in the reference genome, but also by sequencing errors, which make the read less similar to the region of origin, probably, and also by variants, which can make it harder to find the right position of a read. To give you some examples, so for example, let's say this is a sequencing read, and this would be the true position of the read, so where it actually comes from, or where the underlying fragment actually comes from in the genome that has been analyzed. And this could be an alternative position which happens to have exactly the same sequence as here. So now it's unclear where to put the read, right? So and then it depends a lot on the algorithm that is used to do this alignment, whether it correctly detects that or whether it fails to detect that properly, or maybe assigns the read to the wrong location and so on. Then this can be even worse if sequencing errors occur. Let's again assume this is the correct position of the read, so where it actually came from when looking at the molecules. But the sequencing has introduced a T here, so a thymine instead of a guanine, which is actually the true nucleotide in the read at this position, but the sequencing has made a mistake basically here. And now it can happen that just by accident, by this change here, the read suddenly looks more similar to this location where there is already a T in the reference genome, okay? So then the read alignment would mistakenly assign the read to this position and report basically a wrong alignment. It can still be that it still like kind of properly quantifies that this is very similar to this one, so this and this. And then you would get something that's called a mapping quality, which is very weak in such a case, saying that basically it's still very ambiguous between the two. But let's imagine you have not only a single one, but a second sequencing error, then this can get worse again. So this is still a problem, and this uncertainty has to be taken into account by methods that work on those read alignments. And the last case is when you have a certain variant in the underlying individual's genome, like for example, a deletion of these 5 bases here. And then by accident, this variant suddenly turns the fragment that actually comes or should come from here into looking more similar to a different locus in the genome, like here, because here it aligns without any edit operations, like there's no deletion needed here. So suddenly this looks much more likely than this one, although this is the true location, okay? So these are the challenges here. And those challenges depend a lot on the quality of the reference genome, so whether they are harder or less hard to solve. Currently, the latest release of the human reference genome is version 38, which is already quite good, but it still has certain problems, especially in repetitive regions or in regions that are not properly deciphered yet. And therefore, people are nowadays working on pan-genomes. So these are genomes where the variation of a group of representative individuals is included into the reference. And those can at least help with the last source of ambiguity here. Because if this variant, for example, is a common one and that's present in the reference in the sense of that there is like an alternative path that contains this variant, then suddenly the read alignment step can actually see that this is not so dissimilar as it looks like at first sight when looking at this linear reference genome representation, but instead, like there is at least one case where this deletion is already known, so it could estimate that this position is as likely as this position here, for example. So this is where the methods are currently heading towards. Okay, so in summary, one can say the sequencing process and all the analysis steps that come afterwards still have their challenges, even for the most simple kinds of questions. And therefore, it's very important to do benchmarking. And this is so basically there are two kinds of benchmarking I want to talk about in this talk, and the first one is the so-called global benchmarking. This global benchmarking means the following. So first of all, as mentioned, there are lots of challenges and lots of methods and parameters to use and to choose from in the literature. And also the entire field is still continuously evolving, and new methods and improvements of existing methods are presented very often nowadays still. Thus, it's important to kind of benchmark as comprehensively as possible in order to understand what are the individual strengths and weaknesses of methods and parameter combinations. And usually when benchmarking, there are like two options for choosing data sets. One is real data sets and other is simulating data. And of course, both have their validity. In this talk, I want to concentrate on real data sets because they have the advantage of being realistic and covering ideally all kinds of artifacts and errors that you can see with sequencing, whereas simulation always lacks to some degree realism and is never as perfect as being completely like a real data set. The downside of real data sets is, of course, that you are not always entirely sure about the truth, and I will cover that in the coming slides and how to deal with that. So another important aspect with benchmarking is that one should avoid like promoting overfitting. Therefore, it's important whenever you do a benchmarking to use as many benchmark data sets as possible. And the last point is has been really important to us in this project, which is to do the benchmarking continuously and not only once, because as mentioned, these methods evolve so fast that it's not a good idea to just take a snapshot and then consider this realistic for a longer time. So usually this becomes outdated very fast. So one should try to get this continuous with as little effort as possible. So some examples for benchmark data sets that are around and are used in our projects come next. So the most well-known one is probably the Genome in a Bottle data set. Genome in a Bottle is a consortium led by the US NIST, so the National Institutes of Standards and Technologies. And Genome in a Bottle contains a couple of samples. The most prominent one is probably this one, 12878 or HG001. This is just a very well-studied sample from the HapMap project, and then Genome in a Bottle offers some further sets of samples, which are all of them are larger families, so where you don't only have a single sample. I'm proud to say, as the SnakeMake author, I'm proud to say that they actually use SnakeMake for generating these benchmark reference data sets. And the key idea behind Genome in a Bottle is that they take multiple sequencing technologies and multiple variant calling methods to generate some kind of consensus, high quality variants, and also regions where they are very confident that beyond those high quality variants, there are no further variants. And this then you can use for benchmarking your own pipeline, basically, to understand whether you actually properly find the known high quality variants and whether you properly not find any additional variants in those confident regions. Another benchmark data set is the so-called CHM-EVIL data set. It follows a different approach. So this is based on two cell lines, which are both haploid, so they only have a single copy of each chromosome, the CHM13 and the CHM1 cell line. CHM13 cell line is also well known nowadays because this is the basis for the so-called telomere-to-telomere genome assembly, which is like the next upcoming generation of the human reference genome, most likely. And the idea between CHM-EVIL is basically that they took these two cell lines, sequenced them with very high performance and high coverage Pacific Biosciences reads. So those are long reads which have been selected to have particularly high quality in this study. And those long reads were then assembled into like a new reference genome and then mapped back to the known reference genome. And since the two cell lines are haploid, the assembly is particularly simple. And then you even have phased variants in this benchmark data set. So basically, you know whether two SNVs, for example, two single nucleotide variants are on the same chromosome copy or not. And then the test sample for this data set has been generated by basically mixing in the lab the two cell lines in 50-50 ratio and do a normal Illumina sequencing. And the challenge for a variant calling pipeline is then to find all the variants in the right ploidy again, so whether it's heterozygous or homozygous at each position. And for, depending on the use case, you could even benchmark the phasing with that. Okay, another kind of benchmark data set is the seq-C data set. This follows a different approach again. So there, basically, sequencing was conducted by different labs, and each of these different labs used several technologies for the read alignment and the variant calling, and then a consensus was generated from that. So there's no like confirmation by other technologies. This is basically all short read alignment. But the nice aspect of this is that this is not only for germline, but instead this benchmark is also for somatic variants because this was done on a cancer cell line from a breast cancer sample, so that you can also understand about the somatic variants in the data set. So those with fractions less than 50%, for example. Okay, and the final kind of benchmark data set I want to present about is the so-called Seracare somatic cancer reference material. So this is basically a synthetic or let's say in vitro data set where certain variants have been kind of artificially designed, or known variants that are known to be very hard to call have been artificially designed and mixed with the background DNA from some public sample. And by that, you get like a lab mix of those variants together with the background DNA, which is mixed in a way that it can simulate different allele fractions. And this lab mix can be obtained and can be used for sequencing benchmarking experiments. With this kind of data set, you cannot really talk about false positives in your result because, well, you could by comparing with this known genome if everything would be known about this genome. But the most important application of this is to rather just test whether you find those hard to call variants again in the sample by applying your analysis pipeline. And this is what we do currently in collaboration with GHGA in our benchmarking project as well. Okay, so this brings me to the benchmarking project I want to present about here, which is NC-Bench. NC-Bench has been started by the next generation, by the NGS Next Generation Sequencing Competence Network, funded by the DFG, in our special interest group for statistics and computing. At that time, started still by Susanne Motameni. And I've then taken over that later on. And nowadays, this project has extended towards GHGA, which is also contributing, and most recently, the PM4Onco project of the Medizininformatik-Initiative. So the idea of NC-Bench is as follows. So as mentioned, we want to do this global benchmarking, and it's important to do this continuously because methods evolve, and just having a snapshot that was conducted 4 years ago or so is not really helpful later on. So what we have instead engineered is a way to benchmark variant calls basically automated and continuously. And this works as follows. So NC-Bench primarily is consisting of a workflow and a GitHub repository. And this GitHub repository, so GitHub, for those who don't know, GitHub is a version control system that allows to share source code with others and to develop on that source code collaboratively. And this GitHub repository in our case contains the analysis workflow. The analysis workflow for the benchmarking is written in SnakeMake and utilizes a tool called Data Wizard, which we use for visualizing the results of the benchmark. And then what we do basically is that we obtain the truth sets from the different projects that I've just mentioned. So Genome in a Bottle, CHM-Eval, and so on. And then we evaluate given call sets. Those call sets come from different institutes who have taken part in the benchmark, and they are all provided to the pipeline here via Zenodo. So Zenodo is a storage service for scientific data. And then for each call set, we perform certain steps. So we do some atomization. I will come to that in a minute. We stratify the calls, we match against the truth, calculate things like precision and recall, individual false positives and false negatives, and render a report. And this report is then, let's say, collected into a global report for the entire benchmark via SnakeMake, and this is uploaded as an artifact. And then if we are like on the main branch of the repository, we automatically trigger a homepage rebuild. So the repository comes together with the homepage, and this homepage is hosted via GitHub as well. And upon this homepage rebuild, we basically take the report that has been generated here via the pipeline. We fetch that report as an artifact, unpack it, and deploy it to this URL here, so ncbench.github.io. And this URL is hosted by GitHub itself. GitHub has a service called GitHub Pages, which allows to generate, well, web page services directly out of Git repositories. So as you can see, via GitHub as the underlying platform, we can cover the entire process from downloading the truth data sets, downloading the call sets, analyzing the call sets, building the homepage, and uploading everything into like one interactive report on one platform. So there's no need to trigger anything manually whenever some new updated truth is provided or a call set has been updated. This can be run automatically and the homepage will be updated as well. And this is crucial for having this continuity in the benchmarking process because it doesn't need a lot of person hours for maintenance. It doesn't need a local server for running the benchmark analysis and so on. And what we also wanted to show with NC-Bench is that this all works on public infrastructure. So GitHub is free for open source and science. And we just use the free resources by GitHub. And this works pretty well out of the box. Okay. Then, oh, this is an old slide. Sorry for that. But I changed this a bit. But nevertheless, I will just quickly go over parts of this. I just wanted to briefly talk about certain challenges that occur occurred when we designed NC-Bench as an analysis workflow. So basically, this part of the infrastructure that actually analyzes the call sets. A central challenge when trying to compare variant calls of a certain analysis pipeline with a known truth, like whatever the call sets, the data set from Genome in a Bottle or CHM-eval and so on, whatever I presented just previously, is that you actually interpret variants in the same way and that you actually determine whether a certain variant that is reported by an individual pipeline matches one that is known in the truth data set, okay? And this depends a lot on how the individual pipelines represent variants. So what you see here at the top is 4 different variants that are reported by a variant caller called Freebase. And Freebase is in some sense a bit different than some other variant callers in the sense of that it always tries to determine local haplotypes, so longer variants that combine several individual alterations together. And especially with calls like that, but also with other variant calls, you sometimes run into ambiguity in the representation. And depending on which tool you used for representing a variant, those representations can be quite different. And therefore, it's important to normalize stuff before matching between the call set and the truth. Just to give you an example, if we look at this first variant here, this one here, so this haplotype here is replaced by this haplotype. There are different ways to interpret this in terms of what are the individual parts of this, okay? So we can basically perform 2 different alignments of these 2 haplotypes in order to understand what are the differences between the reference and the truth and the variant. So here is the first one. The top here is the reference sequence, and the bottom here is the variant sequence. So you see in the first case, we have basically replaced this, at this position, we have replaced an A with a T. And at this position, we have inserted a T in the variant compared to the reference sequence, okay? However, the same pair of haplotypes can be also interpreted like this here. So here we have inserted a T already here at the beginning. Then all of this matches, and then we have a mismatch here, okay? So depending on how you align these 2 haplotypes, you get different individual operations here, okay? So for example, in the first case, we have this A to T substitution, which is this one here. And then we have, oh, there's even a second one I just missed, the C to A here. And then we have the C to CT here, which is this insertion of the T. Whereas in the second case, we have an insertion first, C to CT, which is the first one here. And then we just have one additional mismatch here, this C to T, okay? So as you can see, it depends a lot on how stuff is interpreted before doing the matching between the truth and the call set, whether you correctly identify false positives and false negatives. And in order to solve this problem, we have decided to normalize both the truth. This is the one I wanted to show. Both the truth and the call set with the same kind of normalization tool. And we have investigated a lot of different versions of BCF tools norm, which is the tool we use for normalization, and also different kinds of benchmark evaluation tools like HAPI and RTG-VCF eval. And in the end, it turned out that a very recent version of BCF tools provide the most stable normalization results and also correct normalization results in terms of providing the maximum parsimony solution in a sense. And RTG-VCF eval was the best solution for actually determining what is a true positive, a false positive, false negative, and so on. Yeah, so then the next challenge was actually calculating the precision and the recall. So in other words, like how accurately does the call set represent the true variance in terms of does it not provide more variance than are actually expected in the truth, and also in terms of recall, does it find all the variants that are supposed to be found by the truth data set. And for the two measures, like the recall and the precision, different behaviors should be, or one would strive for different behaviors. So for the recall, it's fine to expect that it will go down the weaker the evidence is. Whereas for the precision, it's very important that it's independent of the evidence strength, because otherwise, like if the precision goes down, if you have like only a very few reads at a certain position, that would mean that the pipeline can generate lots of false positives if you have a weak sample. Whereas if the precision stays constant, but just the recall goes down, it just means that the pipeline correctly identifies the uncertainty in the data. So remember my first slides about the mapping uncertainty, the sequencing uncertainty, and so on. So that the pipeline correctly identifies this uncertainty, and in that case, the precision would stay constant. And this difference is often neglected by benchmarking projects so far on whole genome or genomic variant calling in general. But we wanted to do it different in NC Bench, so that we decided to always stratify the truth and the call sets by the strength of the evidence at each individual locus, and thereby being able to, let's say, not only give a global recall and precision, but rather do this by evidence strength and thereby show this property of whether the pipelines are actually able to properly assess the uncertainty, and not only whether they give on a binary scale the correct answer, whether a variant is present or not overall over the entire data set. Okay, and then the second decision we made is that we treat the genotype that is reported, so whether the variant is heterozygous or homozygous, or whether it has a certain allele frequency, separate from whether a variant is actually present or not. So for presence and absence of variants, it's important to distinguish precision and recall like this. Whereas for the genotype, it's fine that the precision may decrease with lower read depth, because the genotype is usually just a maximum likelihood estimate. So basically the most likely explanation, whether a variant is heterozygous or not, or whether it has a certain frequency. And that is, of course, much harder when you have less reads. And therefore, we treat the two separately in the analysis. Okay, so I don't know. I think I run out of time already. So just very quickly, this is how, sorry, this is very small. This is how the results of NC Bench look like. So this is exactly the homepage that is rendered automatically upon every update of this GitHub repository I was talking about. Basically the results. So what you see here is a SnakeMake report. So this is generated automatically. We didn't do anything specifically for this analysis. This is just what SnakeMake delivers us automatically. And the report includes now the results. And as mentioned, the results we render via a tool called Data Wizard. And you can then browse this by benchmark data set and by variant type, and we can look into individual items here, like for example, like SNVs for one particular data set. And we can look into this in the table view and like filter stuff or sort stuff differently depending on what we are interested in. And here you see also the stratification. So we have like the different coverage bins here, more than 30 reads, between 10 and 30, between 1 and 10. And you see precision here, and this is a pipeline that works pretty well in terms of being constant in terms of its precision, whereas as expected, the recall can go down with weaker coverage, and can then see the same stuff as a plot as well. So here one would strive for getting as far right as possible for the recall and having a constant curve here. Don't look at the individual items here. For all the pipelines, this is still work in progress and they can all improve still on stuff. But as you can see here, one would strive for constant curves over the different coverages. So each dot here is a different coverage. So from low coverage to medium to high coverage. Okay, and then just very briefly, what we also investigate is false positives and false negatives at individual locations. And the goal of that is to find patterns and try to understand what individual tool combinations or parameter combinations are particularly good for certain loci or for overall, or like where a certain parameter might help or a certain tool might help to get better results at certain positions in the genome. Okay. Yeah, finally, I want to briefly talk about a nice complement to this global benchmarking I was describing because of the following. So first of all, the truth in those global benchmark sets can always be biased towards the used tools, for example, or towards the used approach. But even more important, those data sets are always limited to certain regions where the underlying analysis process has led to some confidence that there are really no more variants than the one that are reported in the truth data set, which means that certain regions of the genomes are just not covered by these data sets. And at that regions, you don't really know whether a certain pipeline might perform completely differently than what the benchmarking suggests. This is especially true for hard to sequence regions like repetitive regions or genes with homologies like HLA genes and so on. And the third aspect, or the fourth aspect, is actually that those benchmarks are really expensive to compute because those are huge data sets and every pipeline needs to run for quite some time to generate the call sets. And therefore, usually the incentive for repeating this a lot is quite low because the burden for computation is quite high. So these get rarely repeated, which means that you also don't have such a fine-grained reporting on those data sets, like on every release of a certain tool or a certain pipeline and so on. This is very hard to organize. Therefore, it's good to complement global benchmarks with what I would like to call local benchmarks. So the idea here is that one would pick some individual variants or non-variants, so loci where you're actually sure that there's no variant. And this is informed by things like prior knowledge, orthogonal validation, or manual inspection. And the upside of this kind of benchmarking is that this is very fast to test because those are only a few reads, and you can easily run this through any pipeline really quickly. And since this is fast and lightweight on the resources, it can also be easily embedded into continuous integration platforms like GitHub Actions. So basically, where GitHub allows to run automatically certain commands on your repository, the same technology that we also use for NCBench. And this runs directly in the GitHub cloud. So basically on every commit to a repository, you can automatically run these tests. And thereby can be very certain that you don't like destroy or make anything broken in your code whenever you change some parameters or some software versions or some algorithms in there. So that can be very handy to have a more fine-grained quality control than what you get with these big global benchmarking projects. In order to make this super easy, we have actually in our own variant caller, Velociraptor, we have introduced an automatic test case generation mechanism. So basically, Velociraptor can be instructed to create individual test cases for individual positions of interests. And that is basically a one-liner, which can be done very easily. And then you get an automatically composed test case. So far, we have collected 145 public test cases that are run through on every commit, and 66 private clinical test cases. So from our local clinical collaborations here, which unfortunately cannot yet be public. But we are currently working on making this entire thing kind of available also to other pipelines and other callers. We do this in the PM4Onco project in collaboration with GHGA as well. And that will hopefully at some point offer also a mechanism for other sites to contribute and to get like a big data sets of these small test cases where the truth is known by manual investigation or orthogonal validation, and thereby offer something that is still lightweight, but nevertheless very comprehensive and a nice way to do further quality control on variant calling. Okay, so to conclude, as mentioned and shown, genomic variant calling is still a hard problem, even for just germline variant calling, but even more so for somatic variant calling. And global benchmarks can help here a lot to understand the performance of method and parameter combinations. However, as methods evolve fast, it's very important to do the benchmarking continuously instead of only once and then relying on that for years. And as we've shown in NC Bench, it's pretty easy to use GitHub, SnakeMake, and Data Wizard to set up such a continuous platform quickly without writing a lot of code. So basically, this just contains the analysis code itself. So for the individual steps of comparing stuff between the call sets and the truth data sets from the benchmark data sets. And everything around is just very little glue code that uses this GitHub platform to do the automation of the individual items. And finally, I would argue for complementing those global benchmarks by local benchmarks. And we're currently working on making that very easy and also being able to collect that on a national scale or maybe even at some point on a global scale at some point, in order to get a large data sets of super fast tests that allow everybody to continuously monitor the performance of tools, methods, and parameters used in pipelines for their projects. Okay, and with that, I would like to thank all the involved collaborations like NGSCN, GHGA, PM4Onco, and all the involved people, like most importantly, Susanne, Gisela, Friederike, Famke, and Bianca, have all contributed to the actual benchmarking itself, and lots of other people who have contributed to one or the other tool or the manuscript that we have written together. Thanks a lot.
Okay, so thank you very much for joining us today. And yeah, thank you very much, Johannes. Thank you very much, our audiences. And thank you to my GHGA colleagues helping me behind. So yeah, have a great day ahead. And we see us in the next GHGA webinar. Bye-bye. Thank you.
Thanks everybody for listening. Thank you.